So for the introduction, first was a fermentation. Fermentation is a natural biological process where microorganisms such as bacteria, yeast, and molds converts into acid, gases, or alcohol. It has been used for 1,000 years to preserve and enhance food and beverage. Why does it matter? The fermentation play a vital role in the food and the beverage industry to improving flavor, texture, and natural value and shelf life of products consumed worldwide every day. So yeah, there is a ancient discovery that we have been using for it for 1,000 of years. So it is for the improving flavor and texture and natural value. So this is where it's going on. So we will talk about of this in a fermentation, food and beverage in industry.
How does fermentation power the food industry? It starts with history. Louis Pasteur proved living microbes drive the process, while Edward Buchner showed that the enzymes inside them do the physical work. Together, they form biochemical transformation. Microbes consume sugars for energy, creating ethanol, lactic acid, and carbon dioxide. This process alters food in three ways: preservation by dropping pH to stop pathogens, flavor by creating aromatic compounds, and nutrition by pre-digesting complex molecules. Let's look at specific types and mechanisms. First, glycolysis. This is the opening stage for all fermentation. It breaks down glucose into pyruvate to extract initial energy. Second is decarboxylation. This step only happens in alcohol fermentation. The enzyme removes a carbon atom to release carbon dioxide gas. Lactic acid fermentation completely skips this step. Third is NAD+ recycling. This is the final closing step for all fermentation. It dumps electrons onto the remaining molecules like acetaldehyde or pyruvate. This clears out NADH, so glycolysis can start all over again. Instead of three separate mechanisms, think of this as three building blocks. Every fermentation type uses glycolysis and NAD+ recycling, but only alcohol fermentation uses decarboxylation to release gas. Glycolysis is basically a sugar splitting process. It takes place entirely in the cytoplasm cell and does not require oxygen. Before you can extract energy from glucose, the cell must invest energy. Now, the cell harvests energy from the two 3-carbon G3P molecules. Every chemical reaction in this phase happens twice, which is once for each G3P. Think of glycolysis as a financial investment. In the first half, the cell spends 2 ATP molecules to destabilize a glucose sugar. In the second half, it reaps the rewards, generating 4 fresh ATP and 2 electron-carrying NADH molecules. The final leftovers are 2 pyruvate molecules, which is served as the raw building blocks for the fermentation pathways. Decarboxylation is the specific biochemical mechanism where a carboxyl group is stripped from an organic molecule and released as carbon dioxide. This process also takes place in cytoplasm cell right after glycolysis finishes. It serves as the bridge between raw pyruvate and the creation of alcohol. Here is the pathway on how it happens. This exact microscopic mechanism is what puts the bubbles in your beer and makes dough rise in the bakery.
Hello everyone, I'm Honey. After glycolysis and carboxylation, one important question remains. How can glycolysis continue when oxygen is unavailable? The answer lies in NAD+ recycling, which is one of the key roles of fermentation. During glycolysis, NAD+ acts as an electron carrier. It accepts electron and is converted back into NADH. However, if all NAD+ is used up, glycolysis cannot continue because there is no NAD+ available to carry more electron. This is where fermentation becomes essential. During fermentation, NADH is converted back into NAD+. The regenerated NAD+ then returns to the glycolysis, allowing glycolysis to continue producing ATP even without oxygen. Therefore, the main role of fermentation is not to produce a large amount of ATP, but to regenerate NAD+ so that glycolysis can continue generating the energy needed for cell survival under anaerobic conditions.
So I will be talking about types of fermentation. First, we have lactic acid fermentation. It carried out by lactic acid bacteria. It convert sugars into lactic acid, give product a sour or tangy taste. Used in mostly yogurt, cheese, kimchi, sauerkraut, and pickles. So we move on to alcoholic fermentation. It's carried out by yeast, convert sugar into ethanol and carbon dioxide. The CO2 makes bird rice. Alcohol give beverages their kicks. It mostly used in beer, wine, bread, and spirits. For lactic acid fermentation, it just basically if we put it as kombucha, they just use sugar and black coffee. They just combine it together. The lactic acid bacteria will eat the caffeine and it will release their the product and give the sour and tangy taste. We will move on to the third is the acetic acid fermentation. So acetic acid fermentation carried out by acetic acid bacteria such as acetobacter is convert ethanol into acetic acid such as vinegar. It produces a sharp or sour flavor used in vinegar, kombucha, and some condiments. The last one is propanoid acid fermentation carried out by bacteria such as Propionibacterium. It's convert lactic acid into propionic acid, acetic acid, and CO2. CO2 gas produced creates the characteristic hole in cheese to give a slightly sweet and nutty flavor. It's used in Swiss cheese and other hard cheese.
Have you ever wondered how everyday products such as yogurt and beer are produced on a large industrial scale? Well, let's take a closer look at how fermentation is applied in this industry. First, let's start with yogurt production, which involves lactic acid fermentation. The process begins with milk preparation and pasteurization. This step is very important because it removes harmful microorganisms and ensures product safety. After cooling, shelter cultures such as Lactobacillus bulgaricus and Streptococcus thermophilus are added. The mixture then undergoes fermentation for about 4 to 6 hours. During this process, the bacteria convert lactose into lactic acid, causing the pH to decrease and giving yogurt its characteristic thick texture and slightly sour taste. Finally, the yogurt is cooled to stop further fermentation before being packaged and stored. Besides yogurts, fermentation is also widely applied in the beverage industry, particularly in beer production. The process starts with barley, which is processed to obtain fermentable sugars during the mashing stage. The mixture is then boiled together with hops to enhance the flavor, aroma, and bitterness of the beer. After cooling, yeast is added and fermentation takes place. During this stage, yeast convert sugar into ethanol and carbon dioxide, producing the alcohol content found in beer. Finally, the beer is filtered, bottled, and prepared for distribution. However, producing fermented product is not only about allowing microorganisms to carry out fermentation. It is equally important to ensure that the process proceeds properly and that the final product are safe. The first method is the pH test, which monitors acidity changes during fermentation. A decrease in pH indicates that microorganisms are actively producing acid and show that fermentation is progressing successfully. Secondly is the sensory testing, which evaluates the taste, aroma, texture, and appearance of the product. This method also helps to detect spoilage or contamination and ensure that the products meet consumer expectations.
Next, kombucha contains mixed microbial community called SCOBY, which stands for symbiotic culture of bacteria and yeast. First, yeast such as Saccharomyces breaks down sugar and produce carbon dioxide. This is the starting point of fermentation. Next, bacteria like Acetobacter convert ethanol into acetic acid and other organic acids. This also forms a cellulose layer known as SCOBY pellicle. Together, this interaction creates a stable system, support fermentation growth, and prevent harmful microorganisms from developing. Moreover, there are several important factors that influence kombucha fermentation. First is temperature, which is usually between 32 Celsius to 30 Celsius. Higher temperatures speed up fermentation and increase acid production. Second is fermentation time, which is typically between 7 days to 10 days. As time increases, sugar decreases while acid increases. This leads to a stronger flavor. Third is microbial composition, which is a diverse mix of microbes help to improve fermentation efficiency. Fourth is oxygen, where kombucha fermentation is aerobic, so oxygen is necessary for bacteria to produce acids. Lastly is pH, where pH decreases during fermentation, which help inhibit harmful microorganisms and stabilize system. Traditionally, kombucha is produced through aerobic fermentation or sweetened tea using SCOBY. In this process, yeast first produce alcohol, which is then converted into acetic acid by bacteria such as Acetobacter. Today, due to increasing consumer demand, kombucha production has expanded using commercial microbial products such as Smart Bev Near and Smart Bev Harvest. This help improve aroma, control acidity, and ensure consistent quality in large-scale production. This shows how traditional fermentation is now combined with modern biotechnology in the food and beverage industry.
In conclusion, fermentation is a natural biological process that plays an essential role in both living organisms and the food and beverage industry. Throughout this presentation, We explored the basic principles of fermentations, its mechanism, different fermentation pathways, production processes, quality testings, and the real-world applications such as kombucha fermentation. Together, these topics demonstrate how fermentation improves food quality, enhances preservation, and supports innovation in biotechnology. Thank you everyone for your attention. That's all from us.